Welcome to the last unit of Clinical Biochemistry Unit 6. Let's start. Hormones and tumor markers. 1. Hormones. Hormones are biochemical messengers secreted by endocrine glands that regulate physiological functions. They can be classified into A. Types of hormones. Peptide hormones, e.g., insulin, growth hormone, water-soluble, act via receptors on cell surfaces. Steroid hormones, e.g., cortisol, estrogen, lipid-soluble, act on intracellular receptors. Amino acid-derived hormones, e.g., thyroid hormones, epinephrine, derived from tyrosine. B. Functions of key hormones. Thyroid hormones, T3, T4, TSH, regulate metabolism. Insulin and glucagon regulate blood glucose levels. Cortisol stress response and metabolism. Reproductive hormones, estrogen, testosterone, LH, FSH, HCG, regulate fertility and secondary sexual characteristics. C. Hormone testing in biochemistry labs. Measured using ELISA, enzyme-linked immunosorbent assay, chemiluminescent immunoassays, and radioimmunoassays, RIA. Used in diagnosing endocrine disorders like diabetes, hypothyroidism, and adrenal insufficiency. 2. Tumor markers. Tumor markers are substances produced by cancer cells or by the body in response to cancer. A. Common tumor markers and their associated cancers. Alpha-fetoprotein, AFP, liver, testicular cancer. Carcinoembryonic antigen, CEA, colorectal and gastrointestinal cancers. Prostate-specific antigen, PSA, prostate cancer. CA125 ovarian cancer. CA19-9 pancreatic cancer. HCG, human chorionic gonadotropin, testicular and gestational trophoblastic tumors. B. Testing methods. Immunoassays, ELISA, RIA. Mass spectrometry. Electrochemiluminescence. C. Clinical applications. Screening and early detection, e.g., PSA for prostate cancer. Monitoring treatment response, e.g., decreasing CEA levels indicate treatment success. Detecting recurrence, e.g., rising CA125 levels suggest ovarian cancer recurrence. Quality control in a biochemistry laboratory. 1. Importance of quality control, QC. Quality control ensures the reliability, accuracy, and precision of biochemical test results, reducing errors in patient diagnosis and treatment. 2 types of quality control. Internal quality control, IQC routine checks using control samples to detect errors. External quality assurance, EQA comparing results with other laboratories to ensure standardization. 3 key QC measures in biochemistry labs. Use of control samples, running known value samples to check accuracy. Calibration of instruments, regular maintenance of analyzers. Standard operating procedures, SOPs ensuring uniform test procedures. Levy-Jennings charts, graphical representation of QC results to detect trends and errors. Westgard rules, statistical QC rules to detect deviations. 4 errors in biochemical testing. Pre-analytical errors, sample collection, handling, and storage issues. Analytical errors, equipment malfunction, reagent issues. Post-analytical errors, incorrect result interpretation or reporting. 5 accreditation and regulatory standards. ISO 15189, International Standard for Medical Laboratories. CAP, College of American Pathologists, CLIA, Clinical Laboratory Improvement Amendments ensure lab compliance. MCQs. 1. Which of the following hormones is secreted by the pancreas and regulates blood sugar levels? A. Thyroxine. B. Insulin. C. Cortisol. D. Estrogen. Answer, B. 2. What type of hormone is cortisol? A. Peptide hormone. B. Amino acid-derived hormone. C. Steroid hormone. D. Protein hormone. Answer, C. 3. Which hormone is primarily responsible for the regulation of metabolism? A. Growth hormone. B. Thyroxine, T4. C. Insulin. D. Testosterone. Answer, B. 4. What is the primary function of the hormone HCG, human chorionic gonadotropin? A. Regulate blood glucose. B. Maintain pregnancy. C. Control metabolism. D. Stimulate muscle growth. Answer, B. 5. Which of the following tumor markers is primarily used for detecting prostate cancer? A. CEA. B. CA125. C. AFP. D. PSA. Answer, D. 6. Alpha-fetoprotein, AFP, is commonly elevated in which type of cancer? A. Breast cancer. B. Liver cancer. C. Lung cancer. D. Colon cancer. Answer, B. 7. CA125 is a tumor marker associated with which type of cancer? A. Ovarian cancer. B. Prostate cancer. C. Brain cancer. D. Lung cancer. Answer, A. 8. What is the role of tumor markers in cancer management? A. Diagnosing infections. B. Monitoring treatment response. C. Measuring body temperature. D. Indicating heart disease. Answer, B. 9. Which laboratory technique is commonly used to measure hormone levels in blood? A. PCR. B. Immunoassay. C. Western blot. D. Hemocytometry. Answer, B. 10. The tumor marker carcinoembryonic antigen, CEA, is commonly used to detect A. Prostate cancer. B. Liver cancer. C. Colorectal cancer. D. Ovarian cancer. Answer, C. 11. What is the main purpose of quality control in a biochemistry laboratory? A. To reduce the cost of tests. B. To improve the speed of testing. C. To ensure accuracy and reliability of results. D. To train laboratory staff. Answer, C. 12. Which type of quality control involves routine checks using known value samples? A. External quality assurance. B. Internal quality control. C. Pre-analytical QC. D. Calibration QC. Answer, B. 13. Which chart is commonly used to monitor quality control trends in a biochemistry lab? A. Line chart. B. Bar graph. C. Levy-Jennings chart. D. Scatter plot. Answer, C. 14. What do Westgard rules help detect in a laboratory? A. Instrument maintenance schedule. B. Quality control violations. C. Sample collection errors. D. Test tube labeling. Answer, B. 15. What is an example of a pre-analytical error in a laboratory? A. Incorrect calibration of an instrument. B. Contaminated reagents. C. Improper sample collection. D. Incorrect test reporting. Answer, C. 16. Which organization sets the ISO 15189 standard for medical laboratories? A. WHO. B. FDA. C. ISO. D. CDC. Answer, C. 17. What is the primary goal of external quality assurance, EQA? A. Verify laboratory performance by interlaboratory comparison. B. Speed up test processing. C. Reduce reagent use. D. Improve staff training. Answer, A. 18. Which of the following is a post-analytical error? A. Wrong patient identification. B. Incorrect sample handling. C. Incorrect test reporting. D. Reagent contamination. Answer, C. 19. What is an example of an analytical error? A. Sample clotting. B. Incorrect instrument calibration. C. Delayed sample transport. D. Mislabeling of samples. Answer, B. 20. What is the purpose of control samples in laboratory quality control? A. To compare results and ensure test accuracy. B. To train new technicians. C. To replace patient samples. D. To increase laboratory profits. Answer, A. Thanks for watching the video. For more videos, subscribe the channel and hit the bell icon to get latest update.